Hello viewers, my name is Damilari. In this topic, we'll be looking at the topic glycolysis and Krebs cycle. In my previous video, I talked about cellular respiration. You get more information from there. But briefly, I want to remind you that cellular respiration simply means the breakdown of glucose to produce energy in the cell, especially in the cytoplasm or mitochondria. So here I'll focus on glycolysis. Take note of the lines I put here. The G and Y means how glucose will be broken down into pyruvic acid. So I'm adding P to this Y. And the SIS you see here represents series. The O here I added 1 meaning 10, meaning that glycolysis has to do with series of chemical reaction, and they are 10 in numbers, such that the glucose will end up as pyruvic acid or pyruvates. So let's look at those 10 chemical reactions, which I call series of chemical reaction by which glucose is broken in to give us pyruvic acid, or you say pyruvates. So the first one, looking at glucose. Remember, glucose is a 6-carbon compound. These are the carbons, 1 here. This points is there's a carbon there. That's 2. So meaning number 1 carbon, number 2. There's another one here 3, another one here 4, another one is here 5, another one here is 6. So it's oxygen that is here. So we can't count this. So we count this carbon that we can see here. Then other points 1, 2, 3, 4, 5. With that one that is there, that's 6. So meaning that this is our glucose. So we say glucose is a 6-carbon compound. So in glycolysis, that glucose will be broken down. And in glycolysis, when glucose is broken down, there will be some enzyme that will make the reaction fast. Remember, enzyme will speed up reaction. So these are the enzymes. These ones are written in blue. They are the enzymes that will speed up the breaking down of the glucose. So the first enzyme is called exokinase. Once you hear exo, from polygon with 6 sides, hexagon, meaning that glucose is a 6-carbon compound. So meaning that the enzyme that will help this 6-carbon compound is called exokinase. And take note, anytime you hear kin, as I keep explaining, because you'll be seeing kin in some places, as you can see it here, kinase. So whenever you hear kin, see another kinase here. So the first thing that should come to your mind is the phosphorus, or you say phosphates, the phosphate group. So how will you remember? Just say KIP, just like in chemistry, Kreb'S apparatus. So once you hear Ki, so that's phosphate. So meaning that this enzyme exokinase will have to take phosphorus, and that's why looking at this stage 1 to 5, they always call it an investment phase. So let me just put I and P. So this is an investment phase. What do I mean by investment? That means they always consume energy, or you say just like if you borrow money. So maybe someone borrows money for investments. So that money he borrows is still going to pay it back. And that's why from 6 to 10 we have payoff. So that's the payoff phase. So we are still at the investment phase where energy is being consumed. So the first energy that will be consumed here is the phosphate, and that's why ATP means adenosine triphosphate, 3 phosphates. So by the time 1 phosphate has been taken, consumed, it will remain 2. 2 means what? Adenosine di. So meaning that from adenosine triphosphate it will become adenosine triphosphate. So that's what kinase has come to do, to borrow 1 phosphate from ATP, which is energy of the glucose. So to borrow 1 ATP, 1 phosphate. So what will they now use the phosphate for? Is bringing it to the glucose. Remember that from the glucose there was CH to OH. So meaning that the H you see here, so it will be leaving. So H will leave this place and the phosphate group will be substituted. So now we can see the phosphate PO3 2-, meaning H has left this place. So and when H is leaving a place, that's an oxidation, and that's why glucose involves oxidation. Remember, when we say respiration, it has to do with oxidation or using oxygen to break glucose. So mean that there's an oxidation here, loss of hydrogen. So what are we saying here? Exokinase involve the using the consumption of phosphate group, which is gotten from the adenosine triphosphate, and then it turns to adenosine diphosphate. So that phosphate group that was consumed here was brought to this glucose by removing the hydrogen there. So now what will now happen, instead of calling the name glucose, you now begin to call the name glucose 6-phosphate, because phosphate has come to the number 6 carbon. If we start our counting from here, we say number 1 carbon, number 2 carbon, number 3 carbon, number 4 carbon, number 5 carbon. So at number 6 carbon, that's where they put the phosphate. Now let's proceed to second stage. This glucose 6-phosphate, as we move to the second phase, which is a reversible reaction, unlike the first one. So from glucose 6-phosphate, you can see fructose 6-phosphate. What does that mean? Remember, glucose and fructose, they are isomer. And what is the meaning of isomer? Something that has the same molecular formula but different structure. So in this case now, it means the enzyme I'll be using is an isomerase, because glucose and fructose, they are isomer. Remember, here we use exokinase, and as I said, that anytime you hear kin, it means phosphate is what we'll be working on. So here now it is isomer we'll be working on. So isomer of glucose is fructose. So instead of glucose 6-phosphate, the name will become fructose 6-phosphate, because the formula of glucose, which is C6H12O6, is also still the formula of fructose. It's also C6H12O6, just that in terms of structure, see glucose here, but look at fructose. It means the fructose is taking the shape of a pentagon having 5 sides, but glucose taking the shape of hexagon. Even by the time you count the carbon, the hydrogen, the oxygen, it will still be the same thing as this. So that's why we call them isomers. So here now, the enzyme here is called phosphoglucose isomerase. Why do we call it phosphoglucose? Because here we have phosphate. That means phosphate of this glucose. That's phosphoglucose. And the isomerase, that means glucose is an isomer of fructose. So that's the enzyme we are using here. So all we just need to do is that we redraw the glucose, change it to fructose structure, leaving the phosphate there. Now we move to step 3. In step 3, we are using another enzyme, which is a kinase. And as I said earlier, once we hear ki, so you remember phosphate. So meaning that here again, you are going to consume another phosphate, and that's why this phase is called an investment phase, or a consumption phase where they will be consuming or borrow. Use this red, you can see that they put red here. It means you have borrowed one phosphate here. Now you want to borrow another phosphate, making 2 ATP you are borrowing. So here now, from this place, from the ATP to ADP, it means another phosphate has been borrowed. Now why are we still using kinase here? So it's because we need another phosphate. So as you can see, this one has only one phosphate, but here now we have 2 phosphate here, phosphate here. So since we have 2, it means the phosphate we borrowed here has been added to this, and the name will change to fructose 1,6-biphosphate. Why? Because fructose structure is still what we are seeing here, fructose. So then the 1,6-biphosphate, what's the meaning of bi? That means the phosphate is in 2 places. So phosphate is here, so at number 1. So it means this time remember we started this one as number 1, so it means there is a phosphate here at number 1. So if you continue, 2 here, 3 here, 4 here, 5 here, then 6 here. At number 6 there is another phosphate, so meaning that 1 and 6, so we have phosphate. So that's fructose 1,6-biphosphate. Now let's move to step 4. In step 4, we are using an enzyme called aldolase. Now what do you need here? The key letter is D. aldolase, that's divide. It means looking at this fructose 1,6-biphosphate, bi, they are 2. It means we can split this thing that is 2 into different halves. That means half on the left, another half on the right. So that's the work of aldolase, dividing this substrate into 2. Now this is the first half here on the left, but this is the second half on the right. How will you know that it has been divided? Remember, there are 6 carbons here, but if you count here, you have 3. 1, 2, 3. The remaining 3 are also here. 1, 2, 3. So meaning the aldolase has been able to divide this fructose 1,6-biphosphate into 2 trioses. Any carbon that is having 3 under glucose or sugar, they call them trioses. So it has been divided into 2 trioses. Now for this first triose, you will notice that this carbon here is having double bond, and before the O, so it's having double bond. Now whenever you see double bond O or carbon double bond O, that's a ketone functional group, or the acanone. So acanone, and that's why you are hearing acetone in the name. So here now we call this one dihydroxyacetone phosphate. So why did they call it dihydroxyacetone? It means from this particular place there is OH here. So OH in the first place. Then this particular thing you are seeing here, you can see PO3 at the end here. So this PO3 is a substitute of hydrogen. So meaning that if we put hydrogen, that's actually OH. So meaning that hydroxy 1, hydroxy 2, that's dihydroxy. Then why is it acetone? The reason is because acetone is for 3 carbon acanone that has 3 carbons, or ketones that has 3 carbons. They call them acetone 3 carbon, because acanone, the simplest acanone is this propanone. So they always call it propan 2-one. So you can even use that idea, because normally anything acet supposed to be 3, 2, because for example acetic acid is 2 carbon. But why is this that this 3 carbon, they now call it acet? So you can just use idea, though that's not the main reason. The idea of, okay, if you want to name this thing, we always call it propan 2-one. So that 2, use it to remember, okay, the 2 represent the acets, so acetone. So, or you say dimethyl acanone, dimethyl, that means methyl on the left, methyl on the right, dimethyl acanone. So use it to remember. So once again, after splitting this, we have our 3 carbon compound here, which is now having the ketone functional group. So, and that's why we said there is OH here, and this one represents OH, so meaning that dihydroxyacetone, the phosphate that is there. So that's dihydroxyacetone phosphate. Now on the right, the second half, so instead of the ketone functional group CO, so no CO here again. Why? Because from this fructose, 1 oxygen has been moved to the left to form the ketone. There is no oxygen to come and form ketone here again. So the only thing we have here is aldehyde. So this one only has CHO, aldehyde functional group. So no ketone functional group here. Okay, now and for aldehyde, you can see that's part of the name given to it. So they gave it the name that has aldehyde because there is CHO there. So, and that's why the name is actually called glyceraldehyde. So you can see it here, glyceraldehyde. So the aldehyde because of CHO that is having. Then the glycer is a 3 carbon. So whenever you see glycer, just like glycerol, propan 1, 2, 3 triol. So propan 1, 2, 3 triol is called glycerol. So now that's where they got the word glycerol there, because it has 3 carbons for glycer. The aldehyde because it has the functional group HCO. So now what did we now notice? Now we have splitted this fructose 1,6-biphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Now why did they put 3 in this one? 3-phosphate because the phosphate is at number 3. If you count carbon, you say carbon 1, carbon 2, and at carbon 3 we see our phosphate. So glyceraldehyde 3-phosphate. Now, what did you see here? They wrote 2 molecules. What does that mean? At this junction, that means as you are moving from stage 5 to stage 6, it means you are moving to another phase, which is the pay-off phase. And take note, at the pay-off phase, there will always be duplicates of anything you do, duplicates. So, and that's why the molecules here will be 2 molecules. Another better way of getting the 2 molecules is that this thing you separated there, they are actually family members, or let me say they are isomers. They are the same. This one here and this one here, making 2. So meaning that the 2 of them, I can call both of them glyceride 3-phosphates, 2 molecules. So, and for your information, this dihydroxyacetone cannot proceed glycolysis, and that's why you can see they put it on the left. So it cannot proceed. So what will now happen? You would rather change it to glyceraldehyde. It will now be double. So now let's proceed. So all we'll be doing now is for glyceraldehyde. Now moving to step 6. In step 6, our enzyme we are using here is called glyceraldehyde 3-phosphate dehydrogenase. What are they adding? Dehydrogenase. Now, what is the work of dehydrogenase? You know, D means to remove. So, and hydrogenase, it means you want to remove hydrogen. So, meaning that from this particular, from this structure, that means from this glyceraldehyde 3-phosphate, I'm going to remove hydrogen. So that's why we call it dehydrogenase. So how will you be able to get that? Okay, look at this place. This our phosphates that we have here. So you can see the phosphates that we have here is still the same phosphates we have here. Now, before we talk of removing hydrogen, look at this C and the O that is here. Remember, there will be extra H here. So look at the C and the O that is also here. Now, where is the extra H here? Is no longer there, meaning that it has been what? Removed. And that's why we call it dehydrogenase. So now that hydrogen has been removed, it means dehydrogenase has done its work. You see that the remaining elements, they are there. So they only removed the hydrogen and they brought in another phosphate. So they brought in phosphate. Immediately they removed the hydrogen. Because of dehydrogenase, they brought in phosphate. And that's why you can see here 2pi telling us that they have added phosphates. And why did they put 2? Like I said earlier, that anything you do now will be double. So meaning that, remember, this glyceraldehyde has been double. We turn this dihydroacetone to another glyceraldehyde. So it means if I do this for the first one, adding phosphate, I have to do it for the second one, adding phosphate. So meaning that I'm adding 2 phosphates. That's why in this step, you will see there is addition of 2 phosphates. Then the hydrogen that was removed, so will be added here. You know, when you remove something from a place, you have to go and add it to another place. So the hydrogen that was removed also will be double. That means 1H was removed here. This is it. From the second glyceraldehyde, another hydrogen was removed. This is it, making 2 hydrogen. Now, that means when we remove them from here, this place, where is it coming to? So it's coming to this NAD. What is NAD? NAD is called nicotinamide adenine diphosphate. Nicotinamide adenine diphosphate. So it has plus. So meaning that the hydrogen that was removed here will be added to that NAD plus. So, and the hydrogen, remember, is double. So one will stay here to join NAD. So NADH. The other one will be brought out as positive. So you say NADH plus H positive. So that's why you can see plus here. So don't forget, this is a coenzyme. NAD is a coenzyme. What is a co? A co is a fellow, like my co-worker, fellow worker. So it means coenzyme, fellow helper, is trying to help the enzyme. All these triols and the likes, they are enzymes. But this one is a coenzyme supporting the enzymes. So, and from the product, take notes, meaning that at the end of glycolysis, if they ask you how many products we have 2 of NADH as our product, we have 2 of hydrogen as our product. So at the end, we'll still get 2 of pyruvate, 2 of ATP. But take note of this, we have gotten 2 of NADH, 2 of hydrogen. So now that this P, as I said, it means they have brought in a new phosphate. They have brought in a new phosphate. Remember earlier we were borrowing the phosphates, but now we want to begin to return the phosphates. So, but we have not even gotten to the payments. We are not yet even paying the money until we get to number 7. So this one is just like invisible phosphates that is being added, or let me say the phosphate that is coming from dehydrogenase. Take note, it's not the phosphate of kinase. You know, I said earlier, when you see kinase, phosphate should come to your mind. But here now, we are seeing phosphates, despite the fact that there is no kinase, but because there is dehydrogenase. So anywhere you see dehydrogenase, also take note that phosphates also work in that line. So that's the phosphate of this. Now we move to, okay, then take note of the name. The name is what? 1,3-biphosphoglycerates. So 1,3-biphosphoglycerates. Now it means the naming of this compound. Why did they say 1,3? It means at the beginning here, number 1, and at the number 3, the third one, number 3, there is what? Phosphate. So meaning that the phospho here 1, phospho here 3. So that's 1,3-biphospho. Now, why did they use the word glycerate? Glycerate from acanoate ester. So because there is a functional group of ester here, and what is the functional group of ester? That's COO. So meaning that the C that you see here and the O that is here and this extra O is for the acanoate. Acanoate. So that's why you see 8. What of the glycer? Glycer. So glycer is always a 3 carbon, 1, 2, 3, like glycerol. So it's a glycerate. So meaning 1,3-biphospoglycerate. So we proceed to step 7. In step 7, our enzyme is phosphoglycerate kinase. Phosphoglycerate kinase. So in this phosphoglycerate kinase, so what are we doing? Remember, anytime you hear kinase, so it means you add P from keep. That means phosphate is coming in. But don't forget for dehydrogenase. So this dehydrogenase is different from aldolase. So don't take this letter D for this one. So this one is dehydrogenase. So it also make use of what? Phosphates. So that's that. Now, if you remember in the investment phase where we are consuming energy, we have consumed 2, this red 1 and 2. So we have consumed 2 ATP. But here now we are seeing green 1, 2, meaning that they are returning the phosphate. And that's why from this you can see the arrow 2 ADP is turning to 2 ATP, meaning that they have restored the ADP, the phosphate. But this time, how are they restoring it? There's 2 here, 2, 2, but from the beginning it was 1, 1. That means they borrowed 1, borrowed another 1, making 2. But here it's paying the 2 immediately. So it's paying the 2 immediately. So means there is 2 phosphates that is that will be represented here. So, and that's why 2 ADP will turn to 2 ATP. Now let's look at the structure. Why do we call it 3-phosphoglycerate and 2 molecules? So we call it 3-phosphoglycerate because at number 3 carbon, this is 1, 2, 3. At number 3 carbon, we can see our phospho. That means the phosphates. So mean that 3-phospho. Then glycer, remember 3 carbon is glycer. Then 8 means acanoate, having COO. So that's for 3-phosphoglycerate. And why did they say 2 molecules? That means this thing I did for the first glyceraldehyde is still what I will do for the second glyceraldehyde. Don't forget we have merged the 2 of them together. So that's why we have 2 phosphoglycerate. Now the next one for stage 8. Okay, so sorry, for stage 7 we said this one was 3-phosphoglycerate. That means at number 3 we saw our phospho, then the glycerate here. So moving to step 8. In step 8 we now said phosphoglyceromutase. Anytime you hear mutase from the name letter M, you can turn it to move. It means the work of mutase is to move, meaning that we are moving the what? The phosphate from the last phase to the what? To the second phase. Instead of calling it 3-phosphol, the name will now become 2-phosphoglycerate. So 2-phosphoglycerate. So that's just the only difference. At number 3 is coming to number 2. And remember, anything we do here will be duplicate. That's why we call it 2 molecules. Now let's move to step 9. In step 9, our enzyme is enolase. So and also water will be removed here. Water will be removed. That's dehydration. So now, why do we call this enzyme enolase, and why do we call this product phosphoenol pyruvate? So from the same structure we have here, something has happened. The hydrogen that was here on top here is no longer here. So the OH that was here is no longer here, meaning that water has been stolen. So that water that is stolen, this is it here, driven out as a loss of water. So water has been stolen. Now, meaning that if we remove the water, this is what will remain. So if we want to name this, so we call it phospho, because there is what? Phosphate there. Phospho what? Enol. Let's start with in. Why do they use the word in? Because here there is a double bond. So there is a double bond here, in. So the double bond came in because as a result of the removal of OH. So this is the in. Then hol. Why did they use the word hol? That means because of the alkanol OH. That means this in came in as a result of the O that was left there. So in O. In came in for O. So that is enol. Then why they use the word pyruvate? A pyruvate, as you can see, this is pyruvate. It is a 3 carbon compounds. 1. Then number 2, it will have a carbonyl, a ketone functional group. 2. And it will also have a carboxylate. That's ester functional group. 3. So from this pyruvate, so it means if you compare it to this, the only difference is that this one has a phosphate. So that's why you can still call it a pyruvate. That means a pyruvate that has the phosphoenol. So that's phosphoenol pyruvate. And as I said, the enzyme enolase. Why did they call it enolase? That means because it's a reaction that is with in and hol. Hol was removed from it. So at any time here ACE means something that is an enzyme. So anything is an enzyme. So just like protease, enzyme of protein, so and so on. Now the last one is number 10, pyruvate kinase. I told you kin is for what? Phosphates. So meaning that, and don't forget, this is a stage whereby they pay off. We are paying back the phosphates that has been borrowed, the ATP that has been borrowed. So we are paying it back. So here, remember, we have paid the 2 of them at the same time here. So meaning that here, trying to release another extra 2 means there is nobody to collect that again because he has cleared his debt. So meaning that the 2 ATP that will be coming out here will be the byproducts. So meaning that we have 2 ATP left. So meaning that at the end of glycolysis, there's always 2 ATP released. So that one is settled. So it means he has paid back his ATP. So it's phosphate. And that's why you do not see phosphate here again. It has been paid. So that's for glycolysis. That's the end of glycolysis. But this also linked to the second one called Krebs cycle. So this glycolysis and Krebs cycle, both of them are actually from what we call aerobic respiration, aerobic, which involves oxygen. So, and this first one we have done takes place in the cytoplasm. So that's where this kind of glycolysis occurs, in the cytoplasm of the cell. But the Krebs cycle, that one will now take place in the mitochondria, the powerhouse of the cell, where there will be more than 2 ATP. You know, here at the end of this, the net ATP is 2. But in that Krebs cycle, it will be 36. As you can see, 36 will be the ATP. If you now add both of them together, it will become 38. So let's continue the work. So that means from the glycolysis, we ended up with the pyruvic acid, or you say pyruvate. And take note, pyruvate is a what? Is a 3 carbon compounds, and here they said there are 2 molecules. Why? Because the glyceraldehyde will double the other time, so we also make the answers to be doubled. And when you double something, 3 carbon in 2 places, that's 6 carbon, which is glucose, if you want to take it back. Now let's now bring this pyruvate 3 carbon down. How will it enter Krebs cycle? Now from the drawing I put here, you notice that I wrote acetyl coenzyme. Acetyl coenzyme, which is a what? A 2 carbon compound. Acetyl coenzyme, so it's a 2 carbon compound. Now let's look at this explanation that is here. So to start with, once you hear Krebs cycle, it is also known as citric acid cycle, or the tricarboxylic cycle. So this is the second phase of the aerobic respiration, and it always makes use of the substrate pyruvate. That is where we stopped the other time. It will use that substrate pyruvate, a 3 carbon compound. It can as well use fatty acid or amino acid. If it's not using that pyruvate 3 carbon, it can use fatty acid or amino acid. So what will it do? We break those pyruvic acid or pyruvate, it will break it and turn it to what? Acetyl coenzyme A. Acetyl coenzyme A. That one is now a 2 carbon compound. So meaning from 3 carbon, it will break it into 2 carbon compound. And how did they get acetyl coenzyme? They added acetic acid with coenzyme, turning it to acetyl coenzyme A. So, and I said it was inside the mitochondria. Now the enzyme that is needed here is the pyruvate dehydrogenase, meaning that as the pyruvic acid or the pyruvate we got in the glycolysis, those 2 molecules of pyruvate we got here, 2 molecules of 3 carbon compound. So as it will be changing or moving down to this place, that means to this Krebs cycle, there must also be an enzyme. Because if you remember here, as we are moving from one stage to another, they were giving us enzyme. So the same way, from this pyruvate to the Krebs cycle, there must also be another enzyme that will be here. So another enzyme. So, and I said the name of the enzyme is what? Pyruvate dehydrogenase. Why? Because we are moving from pyruvate to another compound. Therefore, we are going to dehydrogenase. That means remove hydrogen. So remove hydrogen. And once you are removing hydrogen, there will also be removal of carbon dioxide. Because removal of hydrogen is the same thing as oxidation, and once there is oxidation, that means like the way we breathe in oxygen, there will also be breathing out of carbon dioxide. So that means there will be decarboxylation, and that's why the 3 carbon pyruvic acid or pyruvate will turn to 2 carbon acetyl coenzyme A. Now look at this. This is a coenzyme, nicotinamide adenine diphosphate. Is turning to what? NADH. Why? We have dehydrogenase. We have removed hydrogen from the pyruvates. So since we have removed hydrogen from the pyruvate, it becomes NADH, and the second H will be here because hydrogen is always double. So it comes as a double hydrogen element. So this is the product after adding the hydrogen to this. The hydrogen will be removed from pyruvate. So there's another coenzyme, we call it flavin adenine diphosphate. So this is another what? Coenzyme. So meaning the coenzyme we encountered when we are dealing with Krebs cycle. So we have the NAD and the FADH. So nicotinamide adenine diphosphate and flavin adenine diphosphate. So this also causes decarboxylation. I've said that means carbon dioxide will be released, and that's why it will now reduce the carbon from 3 carbon pyruvate to 2 carbon acetyl coenzyme. So what will this 2 carbon acetyl coenzyme do? It will now go and combine with another compound called oxaloacetic. Oxaloacetic is a 4 carbon compound. So if we add both of them to form to form the citric. So the acetyl coenzyme A, a 2-carbon compound plus oxaloacetic, a 4-carbon compound, will produce citric acid, a 6-carbon compound. So let's now see the structure as it is. Now look at this structure. This is a structure of a Krebs cycle. That means it's going. Krebs is actually a name of a person. So that person was one that explained this Krebs. So now let's see. Remember we were using pyruvate, a 3-carbon compound. So and now as it's coming down, is breaking into a 2-carbon acetic coenzyme A, and I've given the reason because there is a loss of carbon through that decarboxylation, and as a result of oxidation. So that means how oxygen enter. Now we are working with 2-carbon now because one has broken to 2-carbon, and we have noticed how NAD has turned to NADH. So this 2-carbon now, you can see the arrow is coming down, the 2-carbon. At the same time, oxaloacetate is also joining it. So that means this oxaloacetate from the word 8-acanoate. So this is an ester. So joining with this. So the oxaloacetate and the acetic coenzyme, by the time both of them come together, they become citrates. What is the meaning of citrate? From the word citric acid, from orange. So they give us citrate. So this citrate, if you keep doing your cycle, they will begin to break. Some things will be going out, some things will be coming in. So, and that's why there are still 2 other things here. We call it oxidative phosphorylation. Oxidative phosphorylation, and we also have the electron transport. Electron transport. That one is always happening in the complex molecules going on in this same cycle. So in this cycle, you'll be seeing movement, how carbon dioxide will be going out, oxygen will be coming in, and so on. So just take note of what will be produced from citrates to isocitrates to alpha ketoglutarate to succinyl coenzyme A to succinate to fumarate to malate to oxaloacetate. So that's how it will be happening. So, and take note is when succinate is changing to fumarate, that there will be addition of hydrogen, turning flavin adenosine diphosphate to adenine diphosphate to FADH2. So how will you remember all these names? So just use this mnemonics that says OCI. That means officer, can I kill some snakes for medical or medicinal use? So that officer means oxaloacetate. So that's officer. So this is the oxaloacetate, the 4-carbon compound. So officer can, that can is the citrate, the 6-carbon. I means isocitrate. So can I kill is the alpha ketoglutarate. So can I kill some is the succinyl coenzyme A. So some snake is now succinate. So succinyl coenzyme A, some snake. Then they say 4, that's fumarate. Medicinal use, that's malate. So, and take note of the numbers I put beside them. Starting from citrate, they are 6, ketoglutarate, they are 5, succinate 4, succinate 4, fumarate 4, malate 4, oxaloacetate 4, meaning that they actually reduce. And take note, each of them, there is a way they will be releasing ATP, such that at the end they would have released sometimes 36 ATP. So if you add this 36 ATP with the 2 in that glycolysis, it means the total is 38 ATP. And as I said, carbon dioxide will also be produced in the process. And that's why you can see this structure here, saying CO2 going out, hydrogen also being released. So that is how Krebs cycle operates. So thanks for listening.